Hi everyone, today is the second day of my 7-day read an article challenge. In this article challenge, I'm considering scientific articles for my personal mental challenge. You can choose any article of your wish. So eventually after 7 days, I'm supposed to feel better in my mental strength, and we will see how far I reach by that time point. Today is the day 2 of my workout for the mental challenge of reading a scientific article. Today I have chosen a scientific review to go on with you guys. By the end of 7 days, I hope to learn new information, increase my memory function, increase the processing of informations, and make reading scientific articles fun for me. If you take up this challenge, please leave a comment below and let me know how you felt during the challenge and what were the pros and cons of taking this challenge. I will share with you what I have gone through after I have done 7 days of this mental workout, where each day I would read an article. In my case, I'm choosing a scientific article, and after that I will make a presentation out of it. I hope you enjoyed this video, and please leave your comments below here if you like this video or if you dislike the video, and also if you want you can subscribe to my channel. Thank you. Today the paper I'm going to review, it's about electrochromism. The original name of the paper was Electrochromism, a useful probe to study algal photosynthesis, and I have put in the link below the link of the paper for you to study if you would like to go through this paper. However, in this presentation, I'm going to explain to you about electrochromism. Let's begin discussing electrochromism. Before we go into the main topic, we should know that in photosynthetic organisms such as plant, algae, and photosynthetic bacteria, the primary electron donors and the acceptors of photosynthetic complexes are located on opposite sides of the thylakoid membrane. This presence of the electron donors and acceptors on different sides of the membrane causes a proton motive force, which is known as PMF, or an electrochemical proton gradient on the membrane. PMF is represented by the symbol of mu with the subscript of H+, that stands for proton. Since we are looking at the change of PMF, we can represent it as delta mu H+, and it is composed of two main components. These two components are the change in pH, referred to as delta pH, which actually is a representation of the concentration of hydrogen ion present in a moment in either the stroma or lumen of the chloroplast, thylakoids, and the other one is the electropotential gradient, which is the charge dependent component of this equation. The delta pH modulates the rate of electron transfer, and it triggers the de-excitation of excess energy within the light harvesting complexes. The delta psi is required for the metabolite and protein transport across the membrane and induces the shift in the absorption spectra of some photosynthetic pigments present in the chloroplast. This shift in absorption spectra is the so-called electrochromic shift, or ECS, and that is what we are going to learn in this paper. In this paper, we are going to discuss the characteristic features of the ECS and how it is applied in the scientific studies related to photosynthesis. What can we learn by doing the electrochromic shift experiments? In plant cells, there are chloroplasts, within which there are thylakoids present, and in presence of light exposure, there is charge separation that happens across the thylakoid membrane due to the photochemistry of reaction centers that are present within the membrane in the photosystems. These reaction centers are often associated with light harvesting complexes that are known as LHCs, and also charge separation occurs due to electron flow in cytochrome B6F, which is a complex present in the membrane. Now, the activity of cytochrome B6F complex has been discussed by using the Q cycle, and there is transfer of electrons in the low potential chain of this Q cycle. In this presentation, I'm not going to discuss about Q cycle, but if you are interested in knowing more about it, please leave a comment below. So, the flow of electrons across the membrane actually causes the proton motive force. And as I discussed earlier, the proton motive force is composed of the electric field and the proton concentration gradient. This proton motive force that is generated within the membrane actually provides the energy that allows ATP synthesis by the activity of the ATP synthase protein complex present in the membrane. We can study the proton motive force using electrochromism. So now let's discuss what is electrochromism. In order to understand that, first we know that there are photosynthetic pigments present in the light harvesting complexes, and these can absorb light, and due to absorption of light there are defined spectral lines for the excited pigment molecule that is present. These spectral lines can shift or split depending upon the electric field that is provided to them. This phenomenon by which there is splitting of spectral lines is known as the Stark effect, and this happens for photosynthetic pigments as well. Based on this effect, the phenomenon known as electrochromism was first observed by Dusens and reported by them. Now, these pigments that are embedded in lipid membranes undergo a shift in the absorption maxima, because in presence of light, the pigment molecules gain energy. They go from a ground state to an excited state. In order to get back to the ground state, they need to relieve energy. But if they don't relieve energy, in that case, for ground state and the difference of the excited state will provide you a difference in energy. This difference in energy, as you can see on the right hand side of the screen, can be written as a contribution of the change in the dipole moment of the electrons in the ground state versus the electrons in the excited state, along with the electric field that was provided for the splitting effect and the polarizability of these electrons that have moved from the ground state to the excited state. So basically, energy difference is a combination of the dipole moment, the electric field, and the polarizabilities. The frequency by which there will be the shift in the absorption maxima can then be calculated by dividing this energy difference by the Planck constant. The shift of the absorption maxima is the main cause of the band shift effect that leads to the formation of ECS. Now, if this shift of the band is very small, compared to the width of the absorption band, then the equation about the frequency can be rewritten as a differential. So ultimately, the energy difference that we see is what is shown in the graphs that show the ECS spectra, but with respect to the electric field that was provided and with respect to the effect of dipole moments and polarizability that was present during this change of the electron from the ground state to the excited state. So if you look here carefully and minutely in the equation that is on your right hand side bottom of the screen, there is a linear factor associated with the shift and a quadratic factor that is associated with the shift. On the right hand bottom corner is the quadratic shift and the linear one is on the left hand side of the right hand bottom equation that is present. Why it is important knowing about this different kind of linear or quadratic component of the equation, we will come to it in a moment. The electrochromic shift predicts two main features of the electrochromic shift, and these features are based on the equation that we just saw. So the first feature is that the spectrum of the absorption change should follow the first derivative of the absorption band, and its shape should be independent of the field strength. Now look at the figure on the left hand side of this slide. If you see, in absence of electric field, there was a specific absorption spectra shown by the green line. In presence of electric field, this absorption spectra shifted, as shown by the red line. This shift was by the delta nu frequency. Now, for representing the electrochromic shift, we take the first derivative of this absorption change. If you look carefully, this spectra is a Gaussian spectra, right? So if you do a derivative of this spectra, you get a double-waved component. As you can see by the circular line that depicts the field-dependent absorption change that had occurred during the ECS spectra calculation. That's why on the y-axis of the ECS spectra, here we have shown the difference in the energy with respect to the frequency of the shift presented in the x-axis. We can even plot the ECS with respect to wavelengths, as you can see in the bottom two figures over here. On careful inspection of the bottom two figures labeled as A and F, you can see that instead of being a clear Gaussian absorption spectra that we would have assumed it to be, it is composed of combinations of different double-waved components. Why does this happen? Like, why there are multiple double-waved components that are involved in an ECS spectra? The reason for that is because there might be more than one pigment present, each having their separate absorption maxima. And in presence of electric field, there could be different shifts for each of this pigment absorbing at different wavelengths. And that could contribute to the combination of various double-waved components for a ECS spectra. So that's why it is important to deconvolute the ECS spectra in order to get more information, and we will come to it in a few minutes. But based on the example at the bottom of two different organisms having two different ECS spectra, it can be understood that based on the ECS spectra, we can be able to understand that probably the composition of certain pigments have given rise to such spectra. So now if we see an unknown spectra based on where the shifts were for the absorption maxima and based on the deconvolution of the spectra, we should be able to understand the compositions of the pigments present. The second feature of ECS is that the magnitude of the ECS is the sum of the linear and the quadratic functions of the applied field, as I had told you a few minutes ago. And this is based on the equation that is provided on the right-hand side of the screen. In principle, the ECS can be calibrated to provide absolute values of the electric field in the chloroplast. However, this calibration is very hard unless there are both the linear and the quadratic signal present at the same time. On the left-hand side of the screen, you can see the ECS spectra for the linear part of the spectra and the quadratic part of the spectra. In most of the cases, ECS signals have linear response, and linear ECS signal acts as an intrinsic membrane voltmeter, which responds to the changes in the membrane potential. Doing ECS experiments is a good method because we can study the changes in the physiological conditions of photosynthetic organisms in a non-invasive way, whereby we study the changes due to light that affect various functions such as PMF in vivo, that is within the organism. So that's why ECS is a good method. Let us now learn how ECS can show different parameters in photosynthesis. On the right-hand side of the screen, you can see the ECS spectra from various photosynthetic organisms. In vascular plants and in green algae, as shown in the figures on the right-hand side panel, the maximal spectral change that is related to ECS happens at a maxima of 515 nanometer, around that range. The spectral changes at approximately 515 nanometer can be due to the formation of decay of the triplet state of carotenoids. So basically there are a bunch of pigments in the light harvesting complexes, some being the carotenoids and the others being the chlorophylls. Accumulation of specific carotenoids in light exposed plants and green algae lead to the spectroscopic changes at 505 nanometer and 535 nanometer. And these spectral changes are linked to the increase in the pH. If you remember again, pH is an important contributor to the proton motive force that drives the ATP synthesis. Alternatively, ECS signals can be differentiated from other signals by its sensitivity to ionophores. What are ionophores? These are channels through which ions can pass. On passing of ions, there could be changes in the pH or the delta Chi component, and that could affect the PMF again. Previous studies have used this tool to understand where the pigments are located and how the pigments are oriented, because depending upon the location of the pigments and how it is oriented, it will absorb specific light to give absorption maxima, which will be affected by the presence of an electric field that can lead to the change in the absorption maxima, leading to the band shift that gives to the electrochromic shift spectra. The topology of the electron donors and acceptors of PS2 and PS1, which are the photosystems, and the pathway for electron and proton transfer is also important for determining the ECS signal. So generally, for looking into the ECS signal, people have been using repetitive flash spectroscopy. This technique depends on the use of a single turnover xenon or laser flashes to give light signals, and improves the kinetic resolutions so that there is a good kinetic resolution to signal to noise ratio of the ECS, and synchronizes the samples to the environment at which the experiment is being done. In order to understand this ECS signal, it is important that for analysis these signals are correctly deconvoluted, because remember, there could be various pigments having different absorption maxima shift, band shifts that could live in a complex of different curves that are combined together to form the ECS spectra. And we want to see the deconvoluted spectra for the pigment of our interest. From ECS spectra, different kinetic analysis can be done that show how rapid there is excitation and how slowly there is the decay in the excitation of the molecule, showing the ultimate transfer of electrons overall. And for kinetic analysis that are done actually lead in analyzing the PMF. On measuring the kinetics in ECS signal, it has been shown that basically the spectra shows three phases. The first phase is a rapid onset of electric field phase known as the A phase, which is depicted in the red line in the figure A and B. This rapid onset is due to charge separation by the photosystem 1 and 2. The amplitude of the A phase is proportional to the number of active sites in the photosystem, and it can change depending if we reduce some photosystem active site or not. So if you look at the figure B present on the left hand side of the slide, here the A phase initially increases due to presence of both PS1 photosystem 1 and photosystem 2 active sites. But on giving a inhibitor for photosystem 2, the hydroxylamine DCMU combined product that inhibits photosystem 2 now results in a lower amplitude for the A phase, suggesting that A phase is dependent upon the amount of the active sites present for the photosystems in the organism. Their amplitude is dependent upon the active site of the photosystems present and would reflect the stoichiometry between photosystem 1 and photosystem 2 based on the kind of inhibitor that would be used to inhibit either of the photosystems. So what happens after this rapid onset of the electric field that we see in the A phase? This is followed by a slower phase that is known as a B phase, which is represented by the blue line as you can see in figure A. And this reflects the amount of electron transfer that is happening due to the cytochrome B6 activity. So the cytochrome B6F complex is an important component that helps in the electron transfer via plastocyanin and plastoquinone components of the photosynthetic machinery, and the electron transfer that occurs via this complex is represented by the slower phase of the ECS spectra. This phase provides the kinetic information for the cytochrome B6F, and it's used to derive information on the size of the proton motive force in the dark living cells. Following the B phase, there is a decay phase that is represented in the green in the left hand side image, and this reveals the breakdown of the field by ionic flux. This happens when there is hydrogen ion flux through the ATP synthase complex that causes ions to move out from the ATP synthase to produce more ATP, and the variation in the decay lifetime of this phase reveals different levels of the activity of this enzyme. So the measurements of the rate of the relaxation can be understood to estimate how active is the ATP synthase enzyme in effluxing hydrogen ions out from the lumen to the stromal side in order to produce ATP. This actually helps in evaluating the two components of the PMF in the dark. Now let's discuss about the application of the electrochromic shift spectra. Firstly, it can be used to understand the composition of the pigments and how the pigments are oriented. In order to understand this, let's first take a look on the right hand side figure of a blot that was done in order to look at the pigments present in O. tauri organism and Arabidopsis thaliana. Here you can see the PS1 factor and the LHC2 factors of the photosynthetic machinery. As you now know that the light harvesting complexes consist of the pigments, and based on the absorption of the pigments there is change in the electrochromic shift. Now when we look at the graph of the electrochromic shift spectra, looking at the left hand side of the graph shows the blue-green part of the ECS spectrum, and the right hand side of the spectra can be said to be the red region of the spectrum. If we look at the left side of each of these graphs present at the bottom panel, you can see that there are complex features present in the left-hand side, which is also known as the blue-green part of the ECS spectrum, that reflect the combined contribution of different chlorophylls and of carotenoids for this complex feature. The more symmetrical spectra can be found on the other side, that is the red region, and represent the single pigment undergoing the band shift in this region, which is the chlorophylls. In the graph for O Tauri, the blue-green ECS spectrum is carotenoid band shift. That is, the chlorophylls are less sensitive to the applied field in this alga, helping to study the nature and the orientation of the carotenoids. Let's look at the ECS spectra of O Tauri present at the bottom right-hand corner of this slide. The ECS spectra of O Tauri has two major peaks, one at 505 nanometer approximately and the other at 530 nanometer, which is asymmetrically centered on 510 to 515 nanometer, and corresponds to the maximum difference in the absorption spectra of the LHC complexes of Arabidopsis and O Tauri, suggesting that a specific carotenoid is responsible for the electrochromic shift. If you look up on the figure B, on the same side, on the right hand side of the screen, you can see the absorption spectra difference for O Tauri versus Arabidopsis thaliana, and the difference of the absorption plotted from which the electrochromic shift was developed. ECS characteristics thus reflect the environments experienced by the field-responding pigments. In case of O Tauri, those were for the carotenoids. In plants, light-induced generation of an electric field results in the red shift of the spectrum, but blue transitions in O Tauri suggest orientation between the field and transition moment of the carotenoid is antiparallel, implying that the spatial arrangement of the carotenoids within the light harvesting complex must be very different from what is observed in the light harvesting complex 2 of Arabidopsis thaliana. The reason why it is understandable that the transition moment and the spatial arrangement would cause the difference is again based on the equation of the energy difference that is dependent upon the dipole moment and the transition moment of the pigments that are involved in the absorption of the light. Electrochromic shift also helps us to study the change in the photosynthetic apparatus under various environmental stimuluses. How can we use ECS to study different environmental stimulus? Let us take for example what happens during iron starvation. When there is less iron starvation in the soil in which the plant or organism is growing, in that case, the absorption and the uptake of iron is hampered. Now remember, iron is a very important component for the hemes and different iron sulfur clusters present in enzymes that help during the photosynthesis mechanism. If there is less iron, then that will affect the heme centers. So when there is an iron deprivation, photosystem 1 reaction centers that have 12 irons per reaction center have a iron deprivation, and that leads to a decrease of active photosystem 1 centers. This would result in a reduced capacity to reoxidize plastoquinone pools in presence of light. The effect of what happened on the photosystem 1 centers can be visualized by the electrochromic shift when we compare the effect of healthy photosystem 1 centers and photosystem 2 centers from a healthy plant that has proper iron mobilization and uptake occurring versus one from an iron deprivation place, in which case there will be less of photosystem 1 centers present, and that would affect the A phase of the ECS spectra. Kinetic studies, because you can actually visualize the A phase in the ECS spectra kinetic studies, and now because of the less PS1 active sites, there will be less amplitude in the A phase of the kinetic studies. Similar to what we had seen in the experiment that was done with the inhibitors for the PS2 in presence of HA-DCMU complex that reduced the amplitude that was originally for both photosystem 1 and photosystem 2. This kind of experiments that were done on cyanobacteria suggested that decrease of photosystem 1 complex is very severe for the organism, leading to fourfold decrease in photosystem 1 to photosystem 2 ratios. And ECS can estimate the stoichiometry of photosystem 1 to photosystem 2, thus showing how in different environmental stimuli the amount of photosystem 1 to photosystem 2 can vary. ECS can also be used to study if there are alternative electron flow processes leaving the linear electron flow process. Based on the relaxation time differences, there are different signals that are present in the ECS that needs to be deconvoluted based on the relaxation times. Now the rate of the relaxation of triplet of carotenoids is greater than the electrochromic shift decay studied from the kinetic studies. However, the absorption changes at 505 and 535 nanometer for the triplet carotenoids is lesser the electrochromic shift changes that are absorbed. So then this raises a question that where are the electrons flowing? Recently, the techniques have been developed to study electrochromic shift in continuous light. And in order to do so, there has been a method called DIRK, D-I-R-K, where D stands for dark, I stands for interval, R stands for relaxation, K for kinetics, or the dark pulse method have been developed. In this, what happens is under steady state photosynthesis for appropriate wavelengths, ECS signal is measured under continuous illumination, and then the lights are switched off for short time intervals of less than 1 minute. In steady state, the contributions of PS1, PS2 and the cytochrome B6F and of ion leak through the membrane are not distinguishable on the kinetic basis, but can be distinguished when there is a switch to the dark phase. And how can it be done? So if you look at the figure below, the black is the dark phase, the yellow is the light phase. The overall membrane potential is a sum of the photochemical rate of membrane potential formation, the cytochrome B6F specific rate, and the rate of ion leaks. Now, what happens in presence of light? You have all these three components that are present affecting the rate of the overall membrane potential. But as soon as you switch off light and go to the dark mode, now the photochemical rate of membrane potential formation, RPH, becomes 0. So the overall membrane potential is only now dependent on the cytochrome B6F rate and the rate of ion leak. So if you subtract from a ECS spectra of the value of the overall membrane potential slope in the light period versus from the slope in the dark period, you can get the value of the photochemical rate of membrane potential formation. And in this way, you can deconvolute the graph for photochemical rate of the membrane potential form. So this actually helps us to know that if there is an alternative electron flow happening or not. Because in steady state, electron transfer mainly happens from linear electron flow, also known as LEF, in which PS1 and PS2 work in series with similar photochemical rates, which leads to carbon dioxide assimilation. On onset of illumination, Kelvin cycle is inactive, and linear electron flow becomes limited by the low rate of NADPH reoxidation. The photogenerated electrons is then available, leading to different PS1 and PS2 activities. If we study the different rates of the PS1 and PS2 photochemical rates, this will help us to know how is electron diverted away from sink that is not carbon dioxide in vivo, as in case of the steady state situation. And that shows us alternative electron flow systems such as cyclic electron flow. Using the Dirk approach, which allows for illumination followed by brief moments of the dark phase, the fraction of the reaction centers involved in the cyclic electron flow can be studied, along with estimating the membrane potential formation rate due to photochemistry. We can study that by using the formula that is present on the left-hand side of this slide, where RPH was the rate for the photochemical rate of potential. The RPS2 comes from fluorescence measurements following from the equation present on the right-hand side of the slide, based on maximum fluorescence emission, the minimum fluorescence emission in the dark, and the photochemical rate for PS2. From these equations, we can calculate the rate of electron flow via the cyclic electron flow pathway. There is another form of electron transfer that can happen via the water cycle that could generate more ATP funneling electron transfer and is also an alternative form of electron flow, avoiding the linear electron flow system, and is mainly present in algal and cyanobacterial systems. We can study PMF compositions also based on the electrochromic shift approach in presence of light. So what happens during the dark method if there is longer periods of the dark phase that is provided to the system that we are studying? Remember, in the dark method, initial time phases were less than 1 minute. But what if we increase this time seconds? In that case, we get an inverted graph for ECS, which is followed by a initial decrease followed by a slow increase. This you can understand if you look at the right hand side of the slide that shows the light phase followed by an extended dark phase where there is an initial dip and then a slow increase, which is the inverted form of the ECS. This inversion has been interpreted in terms of different rates of relaxation of the two components of the PMF. Generally, after illumination, the relaxation rate for the delta psi component increases over the delta pH. This happens because there is a low dielectric constant in the thylakoid membranes and high hydrogen buffering capacity of the lumen, which slows the rate of charge redistribution along the membrane. When positive charges are transferred from the lumen to the stroma, at that point the PMF is relaxed to its dark adapted level, and that reduces the delta psi component, which also represents the ECS signal in that case, to a level below its dark adapted level, and that compensates for the pH level present. In the second phase, there is a slow relaxation of the pH state, and that would be compensated by the flux of counter ions, which results in the ECS signal to increase. This phase then ends when delta pH and the delta psi component have reached their dark equilibrium state. Based on the overall amplitude of the total and of the inverted ECS signals, attempts have been made to estimate that how much of delta pH or delta psi is present, indicating the size of partitioning of the PMF, and has shown that these estimates give us results showing that the pH is lower than what was initially assumed by using in vitro studies, which leads to the assumption that at a standard lumen pH maintained state of pH 5.7 to 7.8, it can regulate the light capture without damaging the photosynthetic apparatus. However, in the dark, this changes. The PMF across the thylakoid membrane is different, with the delta pH decreasing from 0.5 to 1. Dark plus the light-induced proton gradients in vivo closely resemble the pH gradient estimated in vitro experiments. So this suggests that by using ECS, it should be easy to study the influence of the two components, the delta psi and the delta pH, on the PMF of the membrane. The quadratic component, as discussed in the ECS equation in the very first few slides of this presentation, may help in measuring the pre-existing proton gradients. Generally, there is a linearity between the ECS and the applied field, and that allows measuring changes of the PMF but not its absolute value, which may be required to study ATP synthesis and the regulation of photosynthetic process. However, the ECS signals can be calibrated, leading to the estimate of the absolute value of the electropotential gradient in the light. The amplitude of the linear electrochromic shift response observed upon flash-induced increase of electric field due to charge separation is constant. In contrast, amplitude of the quadratic electrochemical shift response depends on what the value was based on the pre-existing field. Therefore, the amplitude of flash-induced quadratic electrochemical shift when present can be used to understand the absolute value of the electric field. The dark PMF may help maintaining a constitutive amount of activated ATP synthase complex. When the dark PMF helps enzymes involved in the xanthophil cycles in the active state, facilitating the photoprotection during a dark to light transition by speeding up NPQ. And this PMF could be affected by the pre-existing field. The existence of a PMF in the dark could provide the energy source for protein import in the dark, thus maintaining the active photosynthetic apparatus. Thus, the ECS component, when separated in between the linear and the quadratic response, from the quadratic response, we can get the value of the pre-existing field. The value of the pre-existing field will give us the absolute value of the PMF. The absolute value of the PMF helps us to study the ATP synthesis process and thus provides information about the PMF that is present during the dark adapted state of the plant, which is important for the maintenance of the photosynthetic apparatus and in photoprotection mechanism. What I learned from this paper was electrochromic shift is based on the splitting of spectral lines based on the excitation of pigment molecules from ground state to the excited state in presence of electric field. The difference in the energy can be plotted based on the change in the absorption spectra by a specific frequency, which is known as band shift. And from this band shift, we get the electrochromic shift spectral lines, which are basically the first derivatives of the absorption plots for various pigments present in the light harvesting complexes. Since there can be more than one pigment, the presence of various kinds of pigments can give a very convoluted spectrum that needs to be deconvoluted to study the different components involved in photosynthesis. The shape of the spectrum in the electrochromic shift is independent of the field strength. but the magnitude depends on the applied field. By using the electrochromic shift assays, the pigment composition and the orientation effects in a photosynthetic machinery can be studied. ECS can also be used to study the proton motive force composition of the electrochemical potential and the pH composition of the PMF, and how different acclimation environments in presence or absence of lights and other factors can affect the PMF and the overall photosynthesis mechanism. ECS can be studied to know about the existence of the electron flow paths that are there, including the linear electron flow and other electron flow paths such as that done by the cyclic electron flow and the water splitting pathways. And the quadratic function of the ECS, that is not much discussed, does help in understanding if there are pre-existing proton gradients that are already present in the system. So this concludes the day 2 of my 7-day challenge of reading a scientific article. If you like this video or if this video was helpful for you, please put the thumbs up icon, press the bell icon and subscribe to my channel to learn about more articles that I'm going to discuss over the 7 days period. And please share with me your comments on the kind of challenges that you are taking, if you are taking up a 7-day mental challenge of reading an article or not, if you find that it is helpful or not, or how you want to take up a new mental challenge and what kind of articles you are interested in studying. Have a great day ahead and see you till tomorrow. Bye.